Welcome back to my YouTube channel. In this video, I will be explaining PCR, polymerase chain reaction, why it is an interesting technique and what are the steps involved in PCR. PCR is an in vitro technique. That means we perform it in labs. It's a technique which is used to make millions of copies of from a small segment of DNA or a interested gene. For let's say this is the DNA, this short segment of DNA is called a gene. Let's say it is insulin gene. We want to make more millions of copies of that insulin gene. For that we need this PCR technique to make more and more copies of that interested gene. For PCR we need thermocycler. Thermocycler is also called a PCR machine. Then primers are nothing but short sequence of nucleotides, 15 to 20 nucleotides we can see. Then after DNTPs are nucleotide triphosphates like guanine triphosphate, adenine triphosphate, cytosine triphosphate and thymidine triphosphate. These nucleotides are the basis of this DNA. From these nucleotides DNA is formed. Then MgCl. In MgCl, Mg positive is used to which helps the DNA polymerase to add the nucleotides. Then DNA template may, let's say this is the DNA template or interested gene. Then DNA polymerase adds the nucleotides at primers, that I will explain later. Then PCR tubes, and also we need buffers to maintain the pH. Then we add these all these things in a PCR tube. Then add this PCR tube in the PCR machine for further steps. What are the steps of PCR and how to set up the thermocycle or PCR machine? First step is denaturation step, in which we set up the temperature at 94 degree. And as we know, DNA are the two strands are attached with the hydrogen bonding. To break this hydrogen bonding, the 94 degree temperature is very important to break this hydrogen bonding, and we get the two separate strands of that DNA template or a gene. Then annealing step. In annealing, we also called it is also called as primer binding step, in which primer binds here. These are the forward and reverse primers that binds here in these templates, and from here the two strands will form. Then after, there is one of the important step is amplification step, in which millions of copies will form. For example, here are the two primers. Then DNA polymerase, as I discussed earlier, helped by Mg positive, it will add the nucleotides here and form two new strands. From these two strands, there will be four strands here, and two old strands and two new strands, and this chain will continue. This way we will form millions of copies of our interested DNA sequence or a gene. Two strands from two, after binding the primers it will form 4, from 4 it will form 16, from 16 it will form 32, and from 32 it will form millions of copies of that DNA or that interested gene. This was all about the PCR. What are the steps involved in PCR and why it is interesting technique. In next video I will explain this types of the PCR and RT-PCR, which is one of the important type of PCR which is used to detect the COVID patients, whether the person is COVID positive or COVID negative. Thank you for watching.